Welcome to this tutorial, Mastering Clinical and Research Trial Designs, from Observational to Interventional Studies. Clinical and research trial designs are the backbone of evidence-based decision-making in medicine, public health, and science at large. Every piece of medical knowledge we rely on, whether it's the safety of a new vaccine, the effectiveness of a cancer drug, or the association between lifestyle and chronic diseases, comes from studies designed with a clear structure. But here's the challenge. Not all studies are created equal. Some are designed to simply observe patterns in patients or populations, while others are built to test cause and effect by carefully controlling interventions. Understanding how to distinguish these designs, their strengths, limitations, and proper applications is essential if you want to critically read research papers, design your own study, or simply interpret scientific claims with confidence. In this tutorial, we will start by exploring observational designs, often called non-interventional studies, where researchers don't interfere, but only watch what naturally happens. Then, we will move to interventional designs, where researchers actively assign treatments or exposures to study their effects. Along the way, we'll highlight specialized trial designs like randomized controlled trials, quasi-experimental methods, adaptive designs, and more. Before we dive into the details, it's crucial that we all speak the same language. Research methodology has its own set of terms, and if these terms aren't clear, the study designs can sound far more complex than they really are. So, let's begin with the common terms you'll encounter throughout this tutorial. Let's start with one of the most frequently used terms in research, the cohort. A cohort is simply a group of people who share something in common and are followed over time. For example, we might follow a cohort of smokers and compare them to a cohort of non-smokers to see who develops lung cancer. The idea is simple. One shared characteristic, observed over a period. Now, when we follow a cohort, we're usually looking at their exposures, the factors that might influence health. An exposure can be anything: smoking, exercise, diet, a new drug, or even an environmental condition like air pollution. And whenever we talk about exposure, we're naturally interested in the outcome. What happens as a result? Outcomes could be events like developing diabetes, surviving after cancer treatment, or simply a change in blood pressure. When we move from observation into testing cause and effect, we come across the term randomization. This is the process of assigning participants to different groups by pure chance, like flipping a coin. Why do we randomize? Because it helps distribute both known and unknown factors evenly, making groups more comparable. It's what gives randomized trials their strength. But not every study can use full randomization. Sometimes participants are allocated based on availability or physician choice. That's called no full randomization or quasi-randomization. And while it still provides insight, it carries a greater risk of bias. And speaking of comparisons, you'll often hear about a control group. This is the group that doesn't receive the new intervention, but serves as a baseline. It could be a placebo group in a drug trial, or patients receiving standard care in a hospital study. To make these comparisons fairer, researchers sometimes use blinding. This means keeping participants, doctors, or even the whole research team unaware of which treatment is being given. The idea is to minimize bias. In a double-blind trial, for example, neither the doctor nor the patient knows whether they're on the real drug or placebo. Now let's turn to trial objectives. A superiority trial is designed to prove that a new treatment works better than the existing option. By contrast, a non-inferiority trial is not about being better, but about showing the new option is not unacceptably worse. Perhaps it's cheaper, safer, or easier to use. And that's the benefit. Then we have equivalence trials, which go one step further, aiming to show that two treatments are practically the same within a very small margin of difference. All of these designs circle back to one central idea: establishing causality. In other words, proving that one thing truly causes another. But causality can easily be clouded by confounding, which happens when a third factor influences both the exposure and the outcome. A classic example is the link between coffee drinking and heart disease. At first, studies showed a strong association, but it turned out that many coffee drinkers were also smokers, and smoking was the real culprit. This is where we must always be cautious of bias. Bias is any systematic error that distorts the truth of a study's findings. For instance, in case-control studies, patients with a disease may remember their exposures differently from healthy controls. That's called recall bias. Finally, a quick word on validity. Internal validity is about whether the study accurately measures what it set out to within its own population. External validity is about whether the results can be generalized to other groups or real-world settings. In other words, does the study work only in the lab, or will it hold true out in the real world? With these terms in mind, you now have the vocabulary to confidently navigate both observational and interventional trial designs. Next, let's bring these concepts to life by looking at the different types of studies and how they are structured. We will start with the first family of trial designs: observational studies, also called non-interventional studies. These are studies where researchers do not interfere. They don't assign treatments or exposures. Instead, they simply observe what naturally happens, whether in patients, communities, or larger populations. Within observational studies, we have several subtypes, each designed to answer different questions. Let's begin with the simplest: the case report. A case report is a detailed description of a single patient with an unusual or instructive finding. It could be a rare side effect, a novel presentation of a disease, or even a surprising outcome of treatment. For example, imagine a physician encounters a patient who develops a severe skin reaction after using a new cancer drug. By publishing that case, the physician raises a signal that others should pay attention to. Case reports don't prove cause and effect, but they are often the first spark that leads to bigger studies. Now, when more than one similar case is reported together, we call it a case series. Instead of one patient, let's say 10 patients across different hospitals develop the same unusual immune reaction after a new therapy. By grouping them, researchers strengthen the signal that this is not a random event. Like case reports, case series are descriptive. They can't measure risk or prove causality, but they are invaluable for generating hypotheses. Next, we have the cross-sectional study, sometimes called a prevalence study. Think of it as a snapshot, measuring both exposure and outcome at a single point in time. For example, surveying 3,000 adolescents today to ask about physical activity and then recording their current body mass index. This design allows us to estimate how common obesity is and whether it seems linked to exercise levels. The strength is speed and efficiency. The limitation, because exposure and outcome are measured at the same time, we cannot tell which came first, making it weak for causality. Now moving to the case control study. This design works in reverse. Instead of starting with exposure, it starts with the outcome. Imagine we want to study a rare disease, like pancreatic cancer. We gather patients with the disease, the cases, and compare them to similar people without it, the controls. Then we look back to see if cases were more likely to have been. Smoke case control studies are powerful for studying rare outcomes or diseases with long latency periods. They are efficient but they come with challenges. Recall bias, since patients may misremember past exposures, and selection bias, depending on how controls are chosen. By contrast, a cohort study starts with exposure and follows forward to see outcomes. For example, researchers take 10,000 people, half smokers and half non-smokers, and track them for 20 years to see how many develop lung cancer. If this is set up now and followed into the future, it's called a prospective cohort study. If it uses existing records to reconstruct the past, it's a retrospective cohort study. Cohorts allow us to measure incidence, risk, and even multiple outcomes from one exposure. Their strength is establishing temporality, exposure before outcome. Their challenge is cost, time, and the risk of losing participants over follow-up. Finally, in this group, we have the ecological study. This design doesn't use individual level data. Instead, it looks at groups or populations. For instance, researchers might compare national alcohol consumption with liver cancer rates across 20 countries. It's fast and makes use of existing data, but it carries the danger of the ecological fallacy, assuming that what's true for a group is automatically true for each individual. Still, ecological studies are excellent for hypothesis generation and for highlighting broad patterns that deserve deeper investigation. So to summarize, case reports and case series are the first clues. Cross-sectional studies provide snapshots. Case control studies look back from outcome to exposure. Cohort studies follow exposures forward to outcomes. Ecological studies use group-level data to spot trends. Together, these designs form the observational toolbox, the foundation for much of our medical and scientific knowledge. Now that we've seen how researchers can learn by simply observing, let's move on to the next family of designs, where researchers take an active role: interventional studies. Now that we've explored observational or non-interventional studies, let's shift gears to the second major family of trial designs: interventional studies, also known as experimental studies. Here, the researcher doesn't just watch, they take action. They assign participants to receive a treatment, an intervention, or sometimes no intervention, in order to test cause and effect. But interventional studies can be classified in many ways. For our purpose, we'll use two simple methods that make the most sense: by whether the study uses randomization or not, by the objective of the study, whether it aims to prove superiority, show non-inferiority, or demonstrate equivalence. Let's start with the first classification. The strongest design is the randomized controlled trial, or RCT. In these studies, participants are randomly assigned to groups. Randomization makes the groups comparable, balancing out confounding factors, and giving us the clearest window into causality. RCTs come in several forms: parallel group trials. Participants are split into two or more groups, and each group stays with its assigned treatment. For example, one group gets drug A, the other gets placebo. Crossover trials. Each participant receives both treatments in sequence, separated by a washout period. For example, patients receive drug A for 4 weeks, then switch to drug B. This way, each person serves as their own control. Factorial trials. Here, researchers test two or more interventions at the same time in different combinations. For instance, one group gets drug A, another gets drug B, a third gets both, and a fourth gets neither. Cluster randomized trials. Instead of randomizing individuals, entire groups are randomized, such as hospitals, schools, or communities. Stepped wedge trials. A special kind of cluster trial where all groups eventually receive the intervention, but the timing of rollout is randomized. These variations give researchers flexibility to answer different questions. while keeping the power of randomization. Of course, randomization is not always possible. Sometimes it's impractical, unethical, or too costly. In those cases, researchers may still assign an intervention, but without full randomization. These are called non-randomized trials or quasi-experimental studies. OEME common designs include before-after studies, where researchers measure outcomes before an intervention is introduced, and then again afterward in the same group. Interrupted time series, which analyze outcome trends across multiple time points before and after an intervention to see if there's a sudden change in level or slope. Difference in differences, where outcomes in an intervention group are compared with a similar control group over the same time period. These designs can provide useful evidence, especially for public health policies or hospital-wide changes. But because they lack randomization, they are more vulnerable to bias. Now let's consider another way to classify interventional studies, by their objective. A superiority trial is designed to prove that a new treatment is better than the control. For example, a new cancer drug showing higher survival rates compared to chemotherapy. A non-inferiority trial doesn't aim to prove the new treatment is better. Instead, it tests whether the new treatment is not unacceptably worse than the standard. Why? Because the new option may have other advantages. Maybe it's cheaper, safer, or easier for patients to take. An equivalence trial takes it one step further, aiming to prove that two treatments are essentially the same, within a very small margin. This is especially common when testing generic drugs against branded versions. These objectives are important because they define exactly what the study is trying to demonstrate and how the results will be interpreted. Beyond these main categories, interventional designs have some advanced variants. Adaptive trials allow changes to the trial while it's ongoing based on interim results. For example, dropping an ineffective drug arm early. Master protocols include basket trials, umbrella trials, and platform trials, structures that let researchers test multiple questions within a single overarching framework. Pragmatic trials test interventions in real-world conditions, while explanatory trials test under ideal, controlled conditions. N-of-1 trials focus on a single patient, testing multiple treatments in crossover fashion. And finally, single-arm trials, where all participants receive the intervention and outcomes are compared against historical data. These designs reflect how modern research adapts to real-world challenges and scientific complexity. So in summary, interventional studies are classified first by whether they use randomization or not. Randomized trials give us the strongest evidence, while non-randomized trials still provide valuable insights when randomization isn't possible. They are also classified by their objectives, whether to prove superiority, demonstrate non-inferiority, or establish equivalence. And finally, advanced variants like adaptive trials and master protocols push the boundaries of what trial designs can achieve. With both observational and interventional designs in mind, we now have the full toolkit to understand how evidence is generated, interpreted, and applied in clinical and scientific practice. As we wrap up this tutorial on trial designs, I want to thank you for joining me on this journey through both observational and interventional studies. We've seen how some studies are built to simply observe what naturally happens, while others are designed to test cause and effect by carefully assigning interventions. Each design has its place, its strengths, and its limitations, and together they form the evidence base that guides modern medicine and healthcare decision-making. To make this learning easier to revisit, I've also attached infographics highlighting these study designs. Think of them as quick reference tools that you can return to anytime, whether you're reading a research paper, preparing a presentation, or designing your own study. Keep them handy, because understanding trial designs isn't a one-time lesson. It's a skill you'll use throughout your career whenever you interpret or generate evidence. Thank you again for your attention, and I encourage you to explore the attached resources as often as you need.